Niranjani can start. Thangal sir, நான் உங்களுக்கு present பண்றேன்.
Okay, madam.
Okay.
So very warm and pleasant evening to everyone present here. The beautiful thing about learning is that it never ends. It only expands our horizons and deepens our understanding. With that spirit of continuous learning, I warmly welcome you all to day 4 of the 5-day virtual faculty development program on Frontiers in Applied Biosciences and Research Innovation, organized by PG and Research Department of Zoology, St. Mary's College, Autonomous, Thoothukudi. It is indeed a pleasure to see such active participation once again this evening as we continue this academic journey of exploring emerging ideas and research perspectives in the life sciences. A cordial welcome to our resource person, Dave Tangavel, Junior Scientific Officer, Biology Division, Regional Forensic Science Laboratory, Ramanathapuram. To mark the formal beginning of today's session, may I invite Dr. Jessie Dias, Assistant Professor of Zoology, to deliver the welcome address.
Jessie ma'am, your mic is muted. Jessie ma'am, your mic is muted, I think.
Good evening to everyone connected here. It is my privilege, esteemed resource person, Dr. Tangavel, Junior Scientific Officer, Biology Division, Regional Forensic Science, his PhD in Biotechnology from Manoliyam Sundaranar in Tirunelveli. His doctoral research focused on histochemistry, antibacterial farming, and tissue culture studies on threatened endemic medicinal plants. The prestigious BBTO Investigator Award and a major research project funded by the Department of Biotechnology, Government of India, focusing on improved production of bioactive compounds through precursor and agrobacterium-mediated transformation strategies. With over 13 years of teaching experience and more than a decade of research experience, Dr. Tangavel has guided several postgraduate and doctoral students, including MTech and PhD scholars. His research interests include plant conservation biotechnology, in vitro secondary metabolite production, and characterization of bioactive compounds. He is a life member of the Biotech Research Society of India and has contributed extensively to the scientific community through 19 research publications, 2 book chapters, and radio talks on the role of plants in environmental conservation broadcast through All India Radio Tirunelveli. He also served as a reviewer for reputed journals such as African Journal of Biotechnology, Experimental Biology, Journal of Applied Biochemistry and Biotechnology, Journal of Food Science, Microbiology, and Biotechnology. Dr. Tangavel has delivered numerous invited lectures at universities and colleges across India on topics such as plant tissue culture, medicinal plants, plant secondary metabolites, transgenic plants, and advancement in biotechnology. His academic contribution also include participation and presentation in several national and international conferences and seminars. His valuable research contributions and teaching experience have greatly benefited students and researchers in the field of biotechnology. We are indeed honored to have such an eminent scholar and experienced researcher with us today. His expertise and insights will greatly benefit all the participants and inspire young minds to pursue research in biotechnology and plant sciences. On behalf of the organizing committee, I extend a warm welcome to you, sir. I also take this opportunity to express my genial welcome to head of the department, members of the faculty, and all the participants. Once again, welcome you all. Thank you. Thank you.
Thank you, ma'am. Thank you for those warm words of welcome. Let us now take a moment for a virtual group photograph. I request all participants to kindly turn on their cameras so that we can capture this moment of a learning journey. Participants, kindly turn on your cameras. Thank you, everyone. We shall now move on to the technical session of day 4. Today's lecture focuses on a significant topic from forensic biology. With no further delay, I invite Dr. K. Tangavel, Junior Scientific Officer, Biology Division, Regional Forensic Science Laboratory, to share his expertise with us. The virtual platform is now yours.
Madam, let me start.
Yes, sir, you can start.
Is it audible to everyone, madam?
Yes, sir, it is audible.
Okay, madam. Good afternoon, Secretary Madam, Principal Madam, HOD, and all faculty members of the St. Mary's College Zoology Department, and all other faculty friends, those who are joined virtually from different colleges and institutes. So I think almost more than 50% of them will be either directly or indirectly. I'm happy to be here to one among you to share some of my knowledge, but sorry, not will be like presented by our former previous research persons like Ilakya Madam, Kanda sir, Soma Madam, so not like that. So let me place my sincere apologies for making you or giving you somehow a different information rather than paper related or publication related or published articles related works. But whatever I am going to present is whatever the routine job assigned to me and in addition to that, so I will be giving you some inputs as a faculty friends. You may take some insights over such information to promote with some additional upcoming proposals or projects. Very basic technology and instruments in our analysis. But there are several avenues are there and several academics are there to
Excuse me, Tangavel sir. Tangavel sir. Tangavel sir, I have.
Madam,
I think you can either present from your phone or your system. ஏதா ஒண்ணா க்ளோஸ் பண்ணிருங்க சார். அப்ப எக்கோவா இருக்கு. I think it's now closed. Thank you, sir. You can go ahead.
Tell me, madam. Hello.
Sir, you can continue.
Hello. Madam, let me continue. Okay, madam. So, madam, this is our Forensic Science Department logo. Welcome you all. For Tamil Nadu Forensic Science Department, welcome you all. So, the key concept of analysis and all those things are எப்பொருள் எத்தன்மை தாயினும் அப்பொருள் மெய்ப்பொருள் காண்பதறியும். So, I'm very happy to be here with among you people since it is a faculty development program. So I'm happy to be here to have an interactive learning with you also instead of having a training. So let me say it is going to be interactive learning for me with along you people. So madam, I have included some Tamil quotations. Free wants to have the full meaning on the explanations. I will explain. So Katrarmun Kali Uraithal Miha means. So I'm happy to be here with all learned faculty. That's all. And the forensic science, so by applying basic scientific principles with whatever the tools and the methodologies over the suspected articles or items which are being collected from the scene of crime through the police department. So we are assisting to the judiciary and the police department by applying the basic scientific principles in the form of reagents and instruments. And we are providing certain reports. Through that, justice is being established to the victim or deceased. So this is our routine job we are doing. So the basic requirement is a physical evidence. So according to Lockhardt's principle, any contact between any two objects, either they will take or left any energy or in the form of blood stains or footprints. So based on that, we used to move with further. So based on that, we used to move with further investigations. So for every physical imprints or physical evidences, there will be some general characteristics or specific characteristics. So almost like our genus, species characteristics during the taxonomic classifications. So we used to consider such kind of characteristics even for tire marks, footprints, and our DNA fingerprinting also. So for anything and everything we used to follow this Lockhart principle, that is the Lockhart principle of exchange. So any interaction between two objects, either biological or any other chemical objects, that must be having some loss or gain of energy. So these things will be analyzed with an appropriate methodologies. So he is the pioneer in our Lockhart, Edmund Lockhart. So he has established the first forensic lab in Europe. So afterwards, the genesis of this forensic science department including our Tamil Nadu forensic department is like this. So the basic requirements for our analysis is detection kit, EV light. These are all the basic requirements for outdoor crime visits. So in case the specific requirements come to the lab, that is indoor or within our analysis, as far as the biology division is concerned, basic requirements are microscope, spectroscopic analysis, and the TLC, and all other equipments includes the analysis over GCMS. So this is our routine 329 DNSS. So we will be receiving the samples forwarded through Honorable Court, and we suspected articles collected from scene of crime from the police people and submitted to the court. The court will direct us to analyze scientific analysis. So these are the specific questions raised by the Honorable Court. Accordingly, we will test the objects. I will show whatever the objects later slides. So test the objects and we will provide the reports in the. So afterwards, we will have the summons from the court to have the original evidence during the trial. So the tendency of a forensic scientist must be like this. Now, madam, it's audible for everyone?
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
Okay, madam. Okay.
Yes, sir.
So we need to have unbiased mindset while analyzing any suspected articles submitted to the court. That's why I have given this poem, that is quoted from Sirubanjamulam. So again, any one of you want to have any clarifications with this? So in what way this is connected with our routine analysis? So whatever this having a direct connectivity with our analysis, I have given as a quote. Kanvanapu kannotam, kalvanapu sellamai. So this is our right quotation. That is the identity of an analyst or examiner or a scientist should be like that. whatever we have analyzed with an appropriate reagents or appropriate tools and experiments.
Sir, you're not audible, sir. Hello, sir.
To
Tangal
test
not
in the
audible.
form of results. So this result will be
You're not audible,
Now
sir.
it is okay.
Yeah, it's
Yes, sir. It's fine.
Now it is okay, madam.
yes, sir.
Yes, sir.
Okay. So I am not saying that we are helping in a large extent to the judiciary system. So it is a small size help. At the same time, it will be considered as a so that's why I have included this quotation. So this is given by Adivira Ramapalyan, who was one of the famous king in the Pandyan dynasty, as well as he was the best poet also. Yeah, he was ruling some area including Thuthukudi. He has the headquarters in Benghazi. Adivira Ramapalyan, Vetri Verkai. And I will as we are the zoology people. So thing is, the meaning enna madam. So the thing is the tree. You just see the tree. This is a big seed is less than the small egg of a small fish. So though it is such a small seed, it will become a huge tree. It will accommodate this entire king, different armed forces and the people. So likewise, though it is being a small analysis and a report, sometimes it may be considered as a big help. Then as far as our field analysis is concerned, accident cases, we used to go there in site and analyze the damaged area of these vehicles, both these hit vehicles, the four-wheeler as well as the. So we will be searching any part of that particular vehicle might have been subject with such accident. Then in case of suicide cases or suspicious death cases, we will go to the specific site and get some samples. Then in case of non-availability of any poisons and weapons, we used to go through the kitchen and get the unspent food materials and bring to the lab, and we will do it with poison analysis. These are the scene of crime, murder site. We go there and get the sample collections. Then even after the body has been kept in freezers, so sometimes we need to go there and get the sample collections. So this is our routine pattern of getting samples. So these are the suspicious death in our body. So any footprints or blood stains are there. So we people will be having collective effort with the police department also. Then in fire accidents and whether it is an intent firing over this vehicle, such kind of will be analyzing the remnant parts of these vehicles. Then we used to have the collection of these evidences associated with the scene of crime as far as the murder case is concerned. Within these boxes are all so such kind of weapons and materials with blood stains. So likewise, we will be segregating and we will go with an analysis. In case of cases or sexual offense cases, other than the dress material used by the crime. So in addition to that, the medical officers will be directed by the court to get the ear. Accordingly, they will collect and forward it to our lab as per the direction from the court. We will go with an analysis. So for any kind of analysis, we need to have such a tendency of applying an appropriate with a suitable control samples. So that's why I have put this slide. Madam, everyone are accept with this. So for inclusion of this slide with this that map analysis. Madam, audible?
Yes, sir. Yes, sir. Yes, sir.
Okay.
That is audible, sir.
Then let us have a glance. There are several central laboratories, central forensic laboratories that are coming under the Home Ministry, Union Government of India. The first central laboratory established in Kolkata in 1957, and these are the places where central forensic laboratories are there. There is a Central Forensic Science Laboratory in Hyderabad. So likewise, every state government having their own state forensic laboratories, forensic science laboratories. In some other laboratories, they will be attached with directly police department and the judiciary department. As far as Tamil Nadu system is concerned, we are functioning as an independent department, forensic science department. But we are having an association with the police department as well as judiciary department towards the establishment of justice through science. This is our actual vision. And in addition to the laboratories, in every district headquarters, there will be a mobile forensics laboratory with a vehicle. One assistant director will be taking care of the sample collection, SOC, and bringing the samples in time to the lab for analysis. So all those things will be taken care of this mobile forensic lab. So these are all the 10 different regional forensic science laboratories in Tamil Nadu: Coimbatore, Dharmaburi, Salem, Ramanavaram, Tirunelveli, Madurai, Viluppuram, Tanjavur, Trichy, and headquarters in Chennai. So this slide may be invisible, the information may be invisible. Just I wanted to focus you on in the chronology of our Tamil Nadu Forensic Science Department. So next it will be visible if it is zoomed. From 1850s itself, Tamil Nadu Forensic Science from British India itself, it is being functioning. There was a written letter by an analyst to the government secretary for having a better analysis. It needs to be forwarded to some other lab. So just I wanted to make a highlight of the chronology of Forensic Science Department. Then as on now, we are having around 14 divisions in our Forensic Science Department. So one is the Anthropology Division. They will be having a collection of remains and they will go with the whatever the available specimens. Then Ballistics Divisions, bullets and firearms. Then Biology Division. So let us discuss in detail. Blood, semen, saliva, sweat, urine, and all other samples are being subjected with preliminary analysis, including DNA comparisons. Then Serology. So after having the conformity with the blood, we need to have a further confirmation over whether it belongs to A group or B or O. Like in Chemistry Division, petroleum products, oil, and other things. And Computer Forensic Division, nowadays it is being an emerging field of cyber crime. So then there is one more division. Madam, இத கொஞ்சம் ஐடென்டிஃபை பண்ண முடியுமா இது என்ன டிவிஷனா இருக்கலாம்னு? Faculty friends and able to identify. Madam, next slide la answer இருக்கு, but try to identify which division. அடுத்தாட் கொண்ட pronoun. அதுல வர document division madam. Forgery signatures இதெல்லாம் பார்க்கறாங்க இல்லையா? That is one dedicated division, that is document division.
கொஞ்சம் முன்னாடி ஸ்லைட் போக முடியுமா?
நார்காட்டிக்ஸ் சொல்லுங்க மேடம்.
இல்ல சார், முன்னாடி ஸ்லைட் சரியா பார்க்கல. ஓகே சார்.
பார்க்கலாம் மேடம், பார்க்கலாம். எந்த ஸ்லைட்? இதானே மேடம்?
இல்ல சார், இதுக்கு முன்ன அந்த தடுத்தாட்கொண்டன்னு சொல்லி தமிழ்ல இருந்தது இல்லையா?
Please change the slide. The previous one.
How can you go because I'm not tap போட்டு போறோம். No.
Okay, it's okay ma'am, you proceed ma'am, it's okay, no issues.
Okay. And the document division is having such chronological origin, ancient origin from such records. Just I am having a complementary evidence. That's all. Yes. Okay, ma'am. Then DNA division, from parental disputes, the cases are also come with the DNA division. Then explosive division. So nature of explosive, the detonides, the மாதிரிலாம் சொல்றாங்க madam. So likewise, there is another division. Narcotics is ganja, heroin. So for such kind of analysis, there is separate or dedicated division. Then photo and the morphine. Then physics divisions. So they will analyze the footwear and other footprints. Prohibition division. So the cases related with liquors, beer, wine, and prohibited liquors. So likewise, so there are several cases in Tamil Nadu. So they will go with an analysis. Then on one more important division is toxicology division. So they will be concentrating on the poison on the suspicious death cases. So the medical officers will be forwarding the visceral organs. So such organs will be preserved and forwarded to our lab. So our colleagues in that division will go with an extraction of such components and the identification of is there any poison type. Then this is pertaining to our biology division. So the major cases as far as the biology division is murder case, hoax case, and the suspicious death case. So these are all the three major cases we are concerning with biology. Then DNA parental dispute related cases. So after receiving this, so we'll end with the slides. So we'll go with an analysis. This normal vaginal epithelium of a control slide. The bottom one is the again control of spermatozoa. So as and when either one or two spermatozoa is with these epithelial cells, it may be referred as a positive or the sexual offense has been taken place. So such kind of reports we will use it there. Then this is specific antigen kit. That is the basic principle is antigen antibody interaction, almost like our ELISA kit. So there is another kit. So the basic concept is in case of the suspected sample, is there any seminal secretions are there? So ultimately there will be the prostate specific antigen will be there. Such antigen will interact with this antibody. There will be the two lines. So that is the basic mechanism or methodology or principle associated with this test. Then as far as the LOD, that is limit of detection of the samples. Madam, we are all the faculty friends. So you can come up with any new proposals or with further improvement of this methodologies like specific antigen, antibody specific base interactions, because whatever the samples or kits we are having, LOD, the limit of detection or the accuracy is somehow less, madam. So, but still we are using. So because the quality of the sample we are receiving is from crime site, so they will be having very trace quantities of this semen, the saliva, everything. So in case of availability of such a higher methodology,
Sir, your voice is breaking, sir.
No, not audible at all.
Sir, not audible, sir. Sir, you're not audible. Sir, Tangavel, sir. Tangavel, sir, you're not
Priya
audible.
ma'am, he is not in the meet, I think, ma'am. He has some network issue, I believe.
I'll call him. I'll call him. sorry for the inconvenience because maybe he didn't have the network, so he'll join us.
Ma'am, one more request, ma'am. Is there any possibility to share the PPT, ma'am, with the sir's permission?
Sir, maybe we should ask him because, ma'am, it's a government department, no?
Yeah, sure. Understood. That's what I'm asking. With his permission, if okay means you can share. Otherwise, not an issue.
Yeah, sure. Sure, ma'am, sure.
Okay, ma'am.
Dr. Esther, please admit, sir, no?
Ma'am, I've admitted him. I think he's in the meeting.
He's there inside. Tangavel, sir, are you in the meeting?
Crystal formation.
Okay, sir, okay. Yes, sir, you're in the crystal formation slide.
So please continue.
Crystal formation. Madam, this crystal formation will be based on the interaction between the seminal fluids and the fluorescent iodine. So once we put a drop of the fluorescent iodine in a microscopic slide along with the suspected seminal fluid extracted from the vaginal swab, and there will be a possible interaction and precipitation. It will be observed in light microscope. Light microscope, madam. So next analysis is acid phosphatase strips. Acid phosphatase strips so we just place a drop of the suspected vaginal swab extract. In case of positive, the presence of vaginal fluids in that swab, there will be the immediate development of this pink color. Then saliva, that is same salivary amylase recolorization methodology. And we are also having such a kit for salivary amylase, almost like our other PSA, prostate specific antigen. So likewise, we are having the kit for salivary amylase also. Then blood stains. The problem with us, madam, so all the kits and other reagents are working in a perfect manner as and when if they are subjecting with the blood or fresh blood. So in our case, we will be getting the articles arval, kati, dressed. So such kind of articles are partially decomposed, dried, 1 month old, even several months old objects will be receiving. So if we have to trace and search for a small blood stain and dissolve it in buffer, then we have to go with this analysis. This is a small, almost like our ELISA strip. So you can see even such kind of samples. So we could be able to analyze 1:3 lakhs up to 1:3 lakhs dilution, madam. Such kind of dilution. So up to such dilution we can able to identify. So one more arrival with our department is a single step identification of blood as well as the confirmation of the human blood. In earlier days, we used to do 2 different analyses, one for blood and another for origin. So nowadays we are having such a kit with a single step because it is labeled with the human hemoglobin, monoclonal anti-human monoclonal antibody. So it is possible to have such a within 10 to 20 seconds we could able to identify whether the crime site, whether the blood stain, suspected stain, whether it is blood or human blood or not. So such kind of improvements are highly required in our having this kind of analysis in a feasible as well as more accurate manner. So one more important aspect in our analysis, madam, datum. So as far as the suspicious cases is concerned, especially தண்ணிக்கு level இருந்து அந்த இறக்கக்கூடிய cases. So on the suspicious cases, we used to have the collection of sternum, நெஞ்செலும்பு, plus or lungs or part, plus the water sample. Madam, audible to all?
Yes, sir. Yes, sir.
Madam?
Yes, sir. It's audible, sir.
Yes,
Okay.
sir. Yes, sir.
The sternum sample, நெஞ்செலும்பு or part, lungs or part, plus the water sample from the area or water body from where the dead body has been identified. Is to rule out whether this death has occurred the water body exactly or in some other place after this murder or suicide. So they might have been drowned in this site. So for such things, we used to go with nitric acid extraction of this bone marrow from sternum and lungs. Then we will compare the related datum morphological identification with the same group of datum which is being obtained or extracted from this controlled water sample. So this is our comparative study. Again, as I told you earlier, so I will be giving you a small insights to have certain new project proposals. So likewise, though I have not at all been fully permitted to do such kind of research work, so I was trying to have a small piece of research work in a form of research article oriented contribution. So I have got this special permission. Here, if we want to go for publications, we have to get a special permission from our directorate. As per that permission, so as far as this analysis is concerned, so our routine pattern of analysis is nitric acid extraction of this sternum. But in my approach, nitric acid, hydrochloric acid, hydrogen peroxide in different concentration and combination. So for the past 2 years, I was attempting to devise a new methodology out of these attempts. So recently, I could able to have a better density as well as diversity in case of the hydrogen peroxide and hydrochloric acid when they have been incorporated along with this nitric acid. So such kind of analysis or such kind of upcoming methodologies or new project proposals you people can take up, and so these are all the less explored or underexplored areas as far as the forensic analysis is concerned. You can come up with more better results than myself. So this is my request as well as the suggestion for your faculty friends. Then hair analysis. So the hair comparison, the suspected hair from the SOC and the control hairs from the accused or somebody else. These are all our routine methodologies. So always as a faculty friends, as far as the faculty development program is concerned, we need not confined with our, next slide madam, we need not confined with our routine methodologies or routine assigned jobs. So let us go something beyond our routine. So in my aspect, so there was a rare case from forest department. So there was a suspicion whether these two samples, one has been banned from a smuggling group and one is provided as a control sample from the forest department. So they went to the court. The honorable court directed our department to go with an analysis to confirm whether these two wood pieces are belongs to the same tree or not. So very few people have been nominated for this. One among Such people are myself. So the nitric acid and hydrochloric acid and sodium hydroxide combinations. So we have extracted or macerated the wood pieces. Then these wood pieces have been macerated into single fibers. As a result, these single fibers were subjected with microscopy or micrometry. But we have the better micrometry system, software enabled system. We could able to go with a single fiber analysis and compare with certain control samples for want of clarity. So this is not this sample, some other control samples. So all other samples we have compared and this data which we have provided. So based on this data, so we were able to give a report to the court and forest department. So out of this, so myself was capable of getting a special recognition from our directorate exclusively for this small piece of work other than our routine cases like blood cases, POXO cases and all. So likewise, I request all of our faculty friends to contribute little bit more than let us have a special recognitions more than myself. Then respected teachers of zoology department, there are some less explored areas in our next madam, less explored areas in our department, in our forensic sciences. As far as you people for zoology department is concerned, this dead and decayed dead bodies, SOCs, so we could able to identify several type of insects. So this forensic entomology is, on the slides, we don't have such a special division, separate division illa, but if it is going to be there, it will have a better, going to be an indispensable component as far as the present day requirement is concerned. So dead body, next madam, conditions, type of the insect, odor, decomposition, correct madam, odor. With the help of this just formia regina, madam, you people will be knowing that more than myself. So this life cycle and the different stages, so they can give a better clue about these post-mortem intervals. And we need to have such a special or specific type of methodologies to analyze next madam, analyze such a pattern of decomposition, the chronology of this chronology of this decomposition. So for such kind of analysis, model study was undertaken in Huntington, US. To madam, let me summarize the 4 or 5 slides. So slides let me summarize instead of wasting time. So 2 different pigs have been taken for this analysis. They have died almost in the same duration, but they have packed in polythene bags, undisturbed, and they have laid in different places with different climatic conditions. Climatic conditions means the soil microflora is different, temperature, sunlight. So such kind of parameters are different. As a result, there are several analysis has been undertaken. So to summarize this, the maggot was playing a crucial role as far as the decomposition cycle is concerned. Among these maggots, as well as in addition to these maggots, there are several insects and worms could be able to isolated from the decaying body of that pig and other animals subjected with the study. So with this, so the researchers could be able to conclude this life cycle as well as the extract from these insects. So we can be able to identify the poisons and the drugs and all those things. Because these are all going to be the invasive methodologies, we can better promote as well as come out with a better idea as far as the forensic analysis is concerned, with a special reference to decaying body and the special association as time-bound association of the special group of insects is concerned. So next, madam, and our mangi again, it is going to be an insight for you, zoology people. So in Sri Lanka, so several NGO groups in association with these, so they could be able to build the rope bridge for biodiversity conservation of these monkeys and corals. So in our area, let us see such kind of opportunities as a faculty friends or in part of our faculty development program. We could able to see such kind of contribution towards the conservation of biodiversity as well as our career build as far as building as far as the faculty development is concerned. Then some other underexplored or less explored area. Several of our friends will be concentrating or specialized with microscopes. So polarized microscopes are nowadays being subjected with this glass evidences in accident cases and all those things. Then in case of the postmortem samples, the food analysis, the food and the starch on the slide. Food analysis. So with reference to the specific pattern of these starch grains in the
Ma'am, kindly change the slide, ma'am.
Sorry, I am not able to go back.
starch
I think I
grains in
skipped the
this
slide. I'm not able to go back. I
postmortem
think I have to go
samples.
back.
Is it very well possible to make a confirmation over the type of the food the person might have consumed before his death or her death, based on the pattern or based on the nature of the food and the source of the food, the time duration of the food intake, so we can have a better clue. Then for want of having ever any kind of analysis, we need to have different controls. This is my own collection of different hair samples. You might have seen earlier hair comparison. So first I have to rule out whether this hair belongs to human or not. So for that, so whatever you have seen, then the hair sample, madam already காட்டாங்க. ஒவ்வொரு கவர்லையும் இருக்கக்கூடியது ஒவ்வொரு goat, sheep, cow, இந்த மாதிரி samples-ஓட hair collections I have in my laboratory, so that I could able to rule out whether it belongs to human or any other animals. Then we will go with medullary index on the analysis. Then for any kind of such fiber analysis, we need to have the common control system. The same picture, madam, same picture, cotton fiber order, same picture, and the cotton plus silver plus cotton nanoparticles soda, nano composite soda, same picture. Then I have my own collection of plant fibers as well as the graphite crystals லாம் இருக்குது. Then இது some, not my pictures, same pictures. With the help of these pictures, we can able to identify whatever the samples brought by the police department and forwarded by the court, whether it have the resemblance with our earlier samples or not. So this kind of analysis, we need to have such kind of control system. Then soil particle analysis, microscopic analysis of soil, then paint. In case of accident cases, paint analysis, it is possible with the microscope. Then film in accident cases, 2 vehicles. Already, madam, you might have seen the sample collection from lorry as well as the 2-wheelers. So that samples will be subjected with this microscopic comparisons. So based on this comparative or similar features, we could able to identify that bike as well as this lorry might have met with this kind of road accident. So on the Mari analysis, we will be using this kind of microscopic comparisons in our lab. So this is again, if you have any confusion, we can go with the same pictures. Then ஒரு dust particle இருக்கும் மேடம், slide number 115, dust particle. இப்போ SOC போகிறோம், there is no such evidences at all. எந்த evidence 115 மேடம், 115. If there is no such evidences at all, so at least we can have the collection of some dust particles. And the dust particles sort of microscopic analysis itself will yield 115 madam, yield certain clue. So this dust particle belongs to which plant species? Right madam, which plant species? In the plant species in the area, so that upon the dead body on the murder might have been undertaken in that particular area under the canopy of that particular tree or the plant species, and the misplaced on the Mari. Next, so with the help of certain fluorescent stains or reagents, we post-mortem, so we can identify. Then, in one case, madam, there is no evidence in a closed house, a dead body of wife, but there was no clue. Next, madam. There was no clue of their suicide or suspicious death. But while searching, so we could able to identify a dead rat. So on the rat order insects, so we could able to identify this phosphorus poisons with the help of this fluorescent stains labeled with the fluorescent stains and microscopic analysis. So now we can derive a clue about the similar kind of phosphorus poison might have been consumed by these deceased people. So for this kind of analysis, we have to make use of these stains and reagents along with our specialized pattern of microscopes. And this is the pattern of blood stains from weapons and clothes, madam, as far as the murder case is concerned. Then this slide shows, now already Sonamari, we are using very basic methodologies, madam. We are using benzidine method, naphthalene method, then spectroscopic methods, dextrose and NaOH add panni. So we will form a crystal in intermittent crystal with the help of spectrophotometer. We could able to identify the lambda max of hemoglobin. Then in crime scene, so we could able to characterize the distance, duration, and all those things with the pattern of the blood stain. Then serology, saliva, and all those things. Then toxicology, liver, kidney, intestine. So the solvent extraction and distillation. Then we will go with poisons. Then one more routine analysis with our forensic, all forensic lab, madam. This is routine analysis from British India to இப்போ வரைக்கும், madam. The VIPsலாம் வரும்போது ராமேஸ்வரம் இந்த பக்கம் வந்தாங்கன்னா the samples will be given to us. VVIPs, president, vice president, prime ministers அவங்களுக்குலாம். So these are all the different type of poisons are being tested with the food sample which is going to be served to the VVIPs with our again with protocols. So we have to come out with more accurate methodologies. Then as far as the DNA division is concerned, the simple RFLP pattern is being applied to rule out the cases, especially with parental disputes. Madam, so far I was giving you a basic outline about the basic methodologies which we are being used in our laboratory, including our main laboratory, Chennai. But we need not think that they are the limitations. These are all the methodologies are available. Not like that, madam. There are several more accurate methodologies are available in different places, but not in our government analytical laboratories. There are several cases, one in California. So instead of wasting time, I just summarize this case, madam. One case, a mother of a small boy was claiming that, so my boy has been kidnapped and immersed, drowned by some robbers. அப்படின்னு சொல்றாங்க. பட் அந்த பாயோட லங்ஸ் அதில் இருந்த டயட்டம் அனாலிசிஸ் ஆர் நாட் சச் கைண்ட் ஆஃப் கம்பேரிசன். ஏன்னா விமன் herself was wanted to kill her baby. First he was attempting to drown in a fountain in a park, public park. முடியல, பார்த்துறாங்க. அதுக்கப்புறம் ஆஃப்டர் சம் டைம் அப்படியே வாக்கிங் கூட்டு போகிற மாதிரி கூட்டு போய் அந்த பக்கம் இருக்கிறவருடைய ஒரு ஸ்ட்ரீமில் இம்மர்ஸ் பண்ணுறாங்க. பட் as far as the PM sample and datum analysis is concerned, 2 different diversified group of datum கிடைக்குது. அதுக்கப்புறம் அந்த அனாலிசிஸ் வேற மாதிரி போகுது. தென் அந்த லேடி வந்து ஒத்துக்கிட்டாங்க. I myself was attempting in 2 different places அப்படின்னு சொல்கிறாங்க. ஸோ such cases are very interesting. அதுக்கப்புறம் போலன் அனாலிசிஸ், அகைன் இதையும் ஜஸ்ட் ஐ சம்மரைஸ். வேர்ல்டு வார் நடந்தப்போ உங்களுக்குலாம் தெரியும், there was a mass killing of soldiers. So 1994 ஜெர்மனில there was a collection of a huge skeleton. There are 2 hypotheses. One, the skeleton maybe belongs to the victims killed in spring of 1955. The slide, madam. Or another hypothesis, they were the German Democratic Republic nala kollapata Soviet soldiers that may be in June 1953. There are two hypotheses. அந்த ஸ்கல்ல இருந்த டஸ்ட் பார்ட்டிகிள்ஸ் அலாங் வித் சம் pollen grains எடுத்து மைக்ரோஸ்கோப்ல அனலைஸ் பண்ணும்போது அகைன் நம்ம மாதிரி பாட்னி பயாலஜி ப்ரொஃபசர்ஸ் தான் அந்த அனலைசிஸ் taken conducted by academicians only. So they could be able to reveal that almost the pollen grains belong to the plantain, lime tree and the rye grass. They are the prominent plantations during June, July. So with this clue, the conclusion was very easy. So whether this skulls belongs to this 1953 victim of Soviet soldiers, not any other else. So this plant-based pollen grain evidences were there. Likewise, there are several cases are possibly finalized with this kind of information through this plant pollen grains. Then one more, the victim was complaining and accused. He was here with me in this. He was facing the footprints, no footprints and seminal fluids எதுவுமே இல்லை. பட் அங்க ஒரு எவிடன்ஸா கிடைச்சது அந்த பிளான்டோட pollen grains தான். So as a result of this pollen grain analysis, so the coprosma, that is an evergreen shrub, was under that particular area in that garden. The victim might have been subjected to the sexual assault. Then in food poisoning cases, madam, the Clostridium on the picture, food poisoning cases, we will be knowing that food pathogens are more dangerous than any other as far as the food pathogen. The slide on the Clostridium, madam. But the issue is on the culture as well as colony counting. So these kind of methodologies and observations are possible with the E. coli and some other mesophilic organisms like that. But in case of this kind of Clostridium and so we can say VBNC, viable but not culturable groups of organisms. So again, we have to see the possible ways by which so we could be able to come out with the different easy methodologies to have a culture. Once on the culture we could be able to do with ELISA and all those things. Whatever the ELISA which is available, though they are sensitive and the suspected samples நம்ம microbial pathogenic cultures collect பண்ணி அது லைவா இருந்தால் மட்டும்தான் we can go with the enzymatic analysis. அதனால viable but not culturable அந்த issues இருக்கு. But still there are certain attempts made by Miriam in 2020. There is an improved methodology that is known as, madam, audible to everybody?
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir. ELISA, help of this recombinant ELISA, it is possible to have very trace number of such enzymes or this obtained from this Clostridium. Then this is another advanced ELISA, gold labeled immunosorbent assay. Again, it is for detection of Clostridium species jejuni. That is another dangerous food pathogen. The detection limit as far as this improved methodology is concerned, 7.3 CFU. Then instead of going with protein PCR, in our cases, already I have told you, every fresh sample we can do with very little bit quantity of blood, seminal fluid, saliva with other PCR. But as far as the forensic samples is concerned, the nature of samples are very poor. So that's why we need to have multi locations or multi sites where the possible binding of this premise. That's why it is better to have this multiplex PCR. So I think I need not explain the difference between PCR and multiplex PCR. The getting the possibilities of binding as well as the amplification is higher. Right, madam? Next, lamp. Another specific isothermal amplification. Here is also it is possible to have a better yield of this amplicon or the specific. Once this amplicon is more, so the DNA profiling and other RFLP, other further comparisons will be better. Then microarray. So Lee et al. So again for this pathogenic organisms, including Salmonella, this food pen pathogens will be possibly attained or observed with the help of these things. Okay, madam. So I just shown you a very few methodologies by which the forensic samples or whatever the samples which are having poor quality, blood, saliva, semen, and all those things will be subjected with this kind of improved methodologies, but they are not at all affordable to avail these methodologies. Okay, in general, an ideal detection methodology must be satisfy these premier requirements. எல்லாருக்கும் தெரியும் மேடம். There need to have a high specificity, sensitivity, short time, and operational simplicity. Here we have to do all those analysis. We don't have any specific technicians, expertise illa. Single one or two people have to. The most importantly, cost effectiveness. So this is the major barrier as far as the government analytical agencies like us, why we couldn't able to afford this kind of technologies, though they are available in Europe and some other R&D laboratories. That's why in the first one or two slides, there must be an integration of the effort with academicians, forensic analysts, as well as research industries, R&D industries, and law enforcement agencies. Then only we can able to consolidate these devices and more sophisticated methodologies to serve the need, especially of the service of victim. That's why we need to go with SWOT analysis as far as our aim. Our aim is towards justice through science, but we are having this kind of shortcomings, to be frank, shortcomings. Cost effectiveness. So simply our ambition is more, but reality to some extent. Still we are striving to serve because the nature of samples is like this. Whatever the methodologies I just highlighted earlier, ELISA, recombinant ELISA, multiplex PCR, all those things are possible, but in case of these samples still need to be improved. But we are doing with a common PCR, common ELISA, general, analytical, and real methodologies. Methodologies are being supplied to us, but still we are trying to serve in a better extent. So let us have a better interaction, academicians, analysts. So as I told you earlier, I'm not a analyst. Whatever the methodology prescribed by our head office, I have to do. In case of any small deviation in addition to that methodology, I have to get the prior permission. Accordingly, I have got the permission for sternum analysis and datum extraction. So nowadays it is being somehow better. So we need to have an interaction, teaching, reading, as well as conference. So this kind of interaction with analysts like me and researchers like other resource persons, academicians like our Kalidas sir, Ilakya madam, Sopana madam, all other resource persons, the principal, secretary, and all the faculty members of zoology department and other institutions and other faculty friends. So we need to have a better involvement. So everyone will be attending certain training. Let us modified or qualified, but once we have a better involvement, then only everyone will get satisfied. Then let us conclude. So madam, இதுக்கு முன்னால கொடுத்த poem எதுலயும் clarity இல்ல, meaning தெரியலனா let me ask, let me explain. எல்லாரும் கேட்கலாம். But in the last, so I request you, all the faculty members, faculty researchers, and all the concerned, if I hope everyone might have committed to serve to the society, because you are in service sector. Already you have committed, we are happy, but yet we have to commit to serve. So நல்லாவின் பால் முழுதும் கன்றுக்கில்லை, நரம்புவின் மனம் முழுதும் சோலைக்கில்லை, நெல்லாகும் கதிர் முழுதும் நிலத்துக்கில்லை, நிறைகின்ற நீர் முழுதும் குளத்துக்கில்லை, பல்லாரும் கனி முழுதும் மரத்துக்கில்லை, பன்னரம்பின் இசை முழுதும் யாழுக்கில்லை. இது மீனிங் தெரியல மேடம்? ஹலோ,
எஸ் சார்.
அது மாதிரி பசு பூ, all these things are serving the need of others. Likewise, நம் வாழ்வும் பிறக்குழைக்க வேண்டும் வேண்டும். So நம்மளும் let us have whatever the possible means and ways by which we have to serve the society. So I am happy to be here. While I was in teaching, I am serving to the students. Now I am subjected to be here to have a chance of serving to the victim as well as disease. So almost equivalent, or I can say more than equivalent service nowadays I am doing than while I was in teaching service. So you people are serving to the student society. So likewise, every component of our society need to be served by us, either directly or indirectly, whatever the possible means. Then cellular birth, physical birth, social birth. So as biological science faculty, everyone knows that almost all organisms will have these two birth time, cellular birth and physical birth. But the Homo sapiens alone will have the social birth, that is undefined. So let us have an yearly social birth by having the possible service to the society. Thank you, madam. Sorry for the technical as well as the errors from my side, from my laptop. So is there any clarifications? Please get me in touch.
No, sir. No, it was really a wonderful talk. So thank you. Thank you for being with us.
Thank you, sir, for sharing your valuable insights into forensic biology and its role in the analysis of biological evidence. The session is now open for questions and discussion. Participants, you can also post your queries in the chat box.
Okay, madam. So I thank every one of you for giving me this opportunity, especially the principal madam, secretary madam, all the faculty friends of Zoology Department of St. Mary's College, and all the participants connected to Google Meet in this meeting. Thank you, madam. Thank you. Thank you all.
Sir, எனக்கு ஒரு சின்ன டவுட் சார்.
Thanks, sir.
சார், டயாட்டம்ஸ் அனாலிசிஸ் சொல்லியிருந்தீங்க இல்லைங்களா சார்?
ஆமா மேடம்.
அந்த டயாட்டம்ஸ் வந்து எந்தெந்த இடத்துல என்னென்ன மாதிரியான ஸ்பீஷீஸ் இருக்குன்னு நீங்க ஏதாவது லிஸ்ட் முன்னாடியே வச்சிருப்பீங்களா? எந்தெந்த வாட்டர் பாடிஸ்ல வந்து என்னென்ன மாதிரியான ஸ்பீஷீஸ் அவைலபிள் அப்படிங்கறத நீங்க வச்சுட்டு அந்த இடத்துலதான் வந்து நடந்திருக்கு அப்படின்னு சொல்லி கண்டுபிடிப்பீங்களா? எப்படி இல்ல ஒரு இடத்துல நம்ம ஒரு மர்டர் பண்ணி ஒரு வாட்டர் பாடியில வந்து அதை டிஸ்போஸ் பண்ணிட்டு இல்ல அந்த அந்த இடத்துல கொண்டு அதை வேற இடத்துல கூட போடலாம் இல்லையா? அப்ப அதுக்கான ஏதாவது எவிடன்ஸ் நீங்க முன்னாடியே வச்சிருப்பீங்களா? ஸ்டாண்டர்ட்ஸ் மாதிரி இந்த வாட்டர் பாடிஸ்ல இந்த டயாட்டம்ஸ் அவைலபிள் இருக்கும் அப்படின்னு ஏதாவது ஸ்டாண்டர்ட்ஸ் மெயின்டைன் பண்ணிருப்பீங்களா? சாரி சார், எனக்கு இது தெரியல, இது கரெக்டா இல்லையான்னு எனக்கு தெரியல.
கரெக்ட் தான் கரெக்டா மேடம். பட் அந்த மாதிரி கேட்லாக்கோ அந்த மாதிரி ஸ்பெசிபிக்கான கண்ட்ரோல் சிஸ்டமோ இது வரைக்கும் இல்லை. பட் எங்களுக்கு எது கண்ட்ரோல்னா ஒவ்வொரு கேஸ்க்கும் செப்பரேட் கண்ட்ரோல், செப்பரேட் கண்ட்ரோல். இப்போ ஃபார் எக்ஸாம்பிள், இப்போ ஒரு கிணறு அல்லது ஒரு பாண்ட், ஒரு லேக், அங்கிருந்து ஒரு டெட் பாடி எடுக்கிறாங்கன்னு சொன்னா அந்த டெட் பாடி பிஎம் பண்ணி கொண்டு வரக்கூடிய ஸ்டெர்னம், லங்ஸும் பிளஸ் அந்த ஒரு பர்டிகுலர் பிளேஸ்லயே இருந்து எடுத்த வாட்டர் பாடி தான், வாட்டர் சாம்பிள் தான் ட்ரீட் அஸ் எ கண்ட்ரோல். பட் இப்ப நான் எப்படி மற்ற ஹேர் சாம்பிள்ஸ்க்குலாம் கண்ட்ரோல் எடுத்திருக்கணும், அதே மாதிரி வி மஸ்ட் ஹேவ் த கண்ட்ரோல் ஃபார் ஆல் டைப் ஆஃப் டயட்டம் வித் ரெஃபரன்ஸ் டு டிஃபரெண்ட் வாட்டர் பாடிஸ் இருக்கணும். ஹியர் ஆஃப்டர் வி ஹேவ் டு எஸ்டாப்ளிஷ். இது வரைக்கும் நம்ம எஃபர்ட்ஸ்ல அந்த மாதிரி ஒரு எஸ்டாப்ளிஷ்டு கேட்லாக் ஆர் ரெபாசிட்டரி ஆஃப் அந்த டயட்டம் ஸ்பீசிஸ் இன் டிஃபரெண்ட் வாட்டர் பாடிஸ்ல இல்லை. அதுக்கு தான் ஸோ அதர் மெரைன் பயாலஜி பீப்புள் அந்த மாதிரி அவங்க கேட்லாக் இருக்குன்னு சொன்னா we will get the reference from them. இப்போ ஃபார் எக்ஸாம்பிள் இன் அனிமல் சாம்பிள்ஸ்க்கு, அனிமல் ஹேர் இதுக்கெல்லாம் சாம்பிள்ஸ்க்கு very few I have the collections with me.
எனக்கு அதுலயுமே டவுட் இருக்கு சார். ஹேர் வந்து டிஃபரெண்ட் டைப்ஸ் ஆஃப் ஹேர் இருக்குன்னு சொன்னீங்க.
அதான் சொல்றேன் மேடம், லெட் மீ கம்ப்ளீட். அதாவது எனக்கு இன்னும் ஃபர்தரா சாம்பிள்ஸ் வேணும் அப்படின்னு சொல்ற பட்சத்துல ஐ ஹாவ் டு கான்டாக்ட் வித் ஃபாரஸ்ட் டிபார்ட்மெண்ட் பீப்புள். அங்க சென்னையில ஒரு இன்ஸ்டியூட் இருக்கு, இன்ஸ்டியூட் ஆஃப் அட்வான்ஸ்ட் இன்ஸ்டியூட் ஆஃப் வைல்ட் லைஃப் கன்சர்வேஷன் சொல்லி ஹேவிங் டிஃபரெண்ட் ஹேர் சாம்பிள்ஸ், ஸ்கின் சாம்பிள்ஸ், அந்த ரெஃபரன்ஸ் சாம்பிள்ஸ் வந்து அங்க வச்சிருக்காங்க. ஸோ அங்க இருந்து தான் ஐ நீட் டு கெட் தி ரெஃபரன்ஸ் மெட்டீரியல்ஸ். அதே மாதிரி இந்த டேட்டாமுக்கும் in case of such kind of confusions I have to approach the marine knowledge centers and the collections இருந்து I have to get the collections. அதுக்கப்புறம் தான் கம்பேர் பண்ண முடியும் in case of such requirements. பட் இப்போ நாங்க பண்றது வந்து அந்த சாம்பிளுக்கு அந்த கண்ட்ரோல் அதோட முடிச்சுக்கிறோம்.
ஓகே.
Simply speaking, we are not at all having any research
standards.
Analysis மட்டும்தான், அந்த மெத்தடாலஜி, அந்த சாம்பிள் அது மட்டும்தான். But still there must have certain exploration and extended maintenance of control samples as you asked. நீங்க கேட்டது கரெக்ட் தான்.
புரியுது சார்.
கிட்ட அந்த ரெபாசிட்டரி கலெக்ஷன் இல்ல மேடம்.
இல்ல. ஓகே சார். இன்னொரு டவுட் சார், அனிமல்ஸோட ஸ்கின் எல்லாம் வந்து நீங்க சாம்பிள்ஸ் காமிச்சீங்க, எடுத்து வச்சிருக்கோம் அப்படின்னு சொல்லிட்டு ஹேர் சாம்பிள்ஸ். இப்போ நம்மளோட ஹேர் இப்ப சாரி சார் ஹேர் இப்ப நம்மளோட ஹேர்ல வந்து கெராட்டின் வந்து அதிகமா இருக்கும். அப்படி அந்த அனிமலோடது அப்படிங்கிறதுக்கு ஏதாவது ஸ்பெசிபிக்கா ஐடென்டிஃபிகேஷன் இருக்குமா சார்?
நாங்க ரெண்டு பேசிக் பாராமீட்டர் பாப்போம் மேடம். ஒன்னு பிக்மெண்டேஷன்,
ஓகே.
அதுக்கப்புறம் மெடுல்லா, மெடுலரி பேட்டர்ன்.
மெடுலரி, ஓகே.
மெடுல்லா, நான் மெடுல்லா ரெண்டு கேட்டகரி. அதுக்கப்புறம் மெடுலரிலேயே வந்து கண்டினியூஸ் மெடுல்லா, செக்மெண்டட் மெடுல்லா, இதெல்லாம் பார்ப்போம். இது எல்லாமே பேசிக். அதுக்கப்புறம் medullary index, the entire diameter of the shaft versus medulla, இது ரெண்டையும் வந்து ரேஷியோ பார்ப்போம். medullary நம்ம TLC ல RF வேல்யூ பார்க்கறோம்ல, அதே மாதிரி அந்த medullary index பார்ப்போம். medullary index வந்து as far as humans hair is concerned it must be around 0.3 மேடம்.
ஓகே சார்.
இதுதான் நாங்க ஹியூமனா இல்லைன்ற ரூல் அவுட் பண்றதுக்கு. பிக்மெண்டேஷன், ஃபர்ஸ்ட் மார்பாலஜி, பிக்மெண்டேஷன், மெடுல்லா பேட்டர்ன், அதுக்கப்புறம் மெடுலரி இண்டெக்ஸ்.
ஓகே.
இந்த மாதிரி டிஃபரெண்ட் சாம்பிள்ஸ்ல பார்த்துட்டு ஆவரேஜ் வேல்யூஸ் வச்சு அந்த மெடுலரி இண்டெக்ஸ் வச்சு தான் நம்ம ஹியூமனா அனிமலான்னு பார்க்குறோம். அதுக்கப்புறம் ஹியூமனுக்குள்ளேயே கம்பேரிசன்ஸ்க்கு வந்து அந்த மெடுலரி இண்டெக்ஸ்லயே அந்த மெஷர்மெண்ட்ஸ் வச்சு தான் நம்ம மைனர் டிஃபரன்ஸா மேஜர் டிஃபரன்ஸான்னு பார்க்கணும்.
ஓகே சார். சார் ரொம்ப நல்லா இருந்தது சார். தேங்க்யூ சோ மச் சார். இது வந்து எக்ஸலண்ட் அப்படிங்கிற வார்த்தையெல்லாம் இல்ல சார், அதை தாண்டி பிளாஸ்ட் பண்ணிட்டீங்க சார் செஷன். தேங்க்யூ சோ மச் சார்.
தேங்க்யூ மேடம். தேங்க்யூ மேடம். சாரி மேடம், சாரி சார் ஃபார் அந்த சின்ன சின்ன இன்ட்ரப்ஷன்ஸ் இருந்தது. மேடம் என்னோட ஸ்லைட்ஸ் இருக்கு. ஸோ as and when you like you can have the slides அந்த PPT இருக்கு, வாங்கி பார்த்துக்கலாம். ஓகே சார். பாலச்சந்தர் சார், எல்லாருக்கும் வணக்கம். இலக்கிய மேடம்,
Thank you, sir.
மேடம் லிங்க்ல இருந்தாங்க, பட் ஐ எம் நாட் ஷூர் இப்ப இருக்காங்களான்னு தெரியல. காளிதா சார், சோபனா மேடம், ஆல் அதர் ரிசர்ச் பர்சன்ஸ், ஆல் தி பார்ட்டிசிபென்ட்ஸ் எல்லாருக்கும் ரொம்ப நன்றி மேடம். வணக்கம். ஓகே மேடம். தேங்க்யூ.
Thank you, sir, for patiently clarifying the queries and for valuable discussion and the usage of Tamil literature to make the topic more easily understandable and more interesting. As we conclude today's session, I request Dr. J. Shobha, Assistant Professor of Zoology, to deliver the vote of thanks.
Thank you, sir.
Okay, madam. Okay.
Thank you, sir.
Thank you.
Good evening to everyone. On behalf of the Department of Zoology, I extend my heartiest gratitude to all those who have contributed to the resounding success of this faculty development program. First and foremost, I would like to express our fundamentals and applications in forensic biology has greatly enriched our knowledge and provide us with a deep understanding of how biological evidence plays a crucial role in modern forensic investigation. The depth of your knowledge and the practical insights you shared have undoubtedly added a few new dimensions to our professional experts. Thank you, sir, for your time and for inspiring with your words. I deeply grateful to our secretary, Reverend Sister Dr. S. Kulandidares, our principal, Reverend Sister Dr. Jessie Fernando, Reverend Sister Dr. M.S. Elilarasi, Deputy Principal, and Reverend Sister Dr. R.O.K. Jessis Alphonse, Director, SSC, for their unwavering support and vision. Their constant encouragement to organize such academic programs allows us to stay at the forefront of educational excellence. A special mention goes to our head of the department, Dr. P.J. Joslin, head and associate professor, and the entire organizing team. An event of this scale is the result of weeks of meticulous planning and teamwork. I thank my colleagues for their tireless efforts in ensuring everything from logistic to technical coordination ran seamlessly. I would like to thank the participants, my fellow faculty members from various institutions. Your active engagement, thought-provoking questions, and enthusiasm are what made this program truly interactive and meaningful. In conclusion, I thank everyone once again for being a part of this journey, continuous learning. I hope we all carry the insights gained today back to our classrooms. Thank you all. Have a wonderful day ahead.
Thank you, ma'am, for those gracious words of gratitude.
Thank you, madam. Thank you, madam. Thank you all.
Thank you, sir. Thank you so much, sir.
Thank you, sir. Thank you, sir.
So I kindly request all the participants to take a moment to fill the feedback link. The link from it has been shared in the chat box. So with this, we gently draw the curtains on today's session. I sincerely thank all the participants for your presence and active participation. We look forward to meeting you all again tomorrow for the final day of this FDP with another enriching session. Until then, thank you and have a wonderful evening.
Thank you, Christina ma'am. Christina ma'am, if possible, kindly share the PPT as well as sir's contact number or mail ID, ma'am, if possible.
Sure, yeah, sure, ma'am, we'll do it.
Yes, ma'am. Thank you so much, ma'am. It was very good organizing, ma'am. Thank you. We learned a lot.
Thank you so much, ma'am.
Thank you.